Good morning. It's August 5th, and this is your daily brief in science. Here's everything you need to know. A major conservation push is underway as Leonardo DiCaprio and the Bezos Earth Fund unveil the Phoenix Species Project, with a $200 million pledge to recover about 100 critically endangered species across mammals, birds, amphibians, reptiles, fish, invertebrates, and plants in 30 countries. This initiative is designed to sustain and scale successful conservation work over the long term, moving beyond one-off grants to deliver enduring impact. It grew from collaborative talks among local governments, environmental groups, and indigenous communities to better coordinate efforts and reverse declines of vulnerable species. The report comes from Los Angeles, with Associated Press collaboration, and IUCN-SSC serves as the knowledge partner to ensure scientific rigor. Population recovery and threat reduction will be tracked with updates on the project's site and social channels. The article notes related biodiversity finance news, including a World Bank-backed Caspian Sea biodiversity project, to provide broader context. Public enthusiasm is reflected in statements from Lauren Sanchez Bezos, who highlights a child-friendly narrative to inspire protection of rare species, while placing the effort within broader science advocacy and funding challenges for science journalism. The project frames a shift from declining trends to potential rebounds through sustained investment, local leadership, and robust recovery actions, aiming to spark a global movement that attracts broader nature investment. Key quotes emphasize that the funding and collaborative platform change what is possible for safeguarding endangered species. Organizers hope the initiative will mobilize other philanthropic donors to join in addressing global biodiversity loss. Tiana Andriamanana of Association Fanambi notes that the funding and global platform enable sustained protection beyond previous budgets, marking a significant shift in conservation capacity, while another quote stresses that the funding allows continuation and expansion of protection where budgets were previously limiting. A new study calls into question NICE's current breast cancer referral criteria for women under 49, showing they may miss up to 95% of cancers that would develop within 10 years. The research compares NICE criteria with Boadicea-based strategies, which improve sensitivity but require more of the population to undergo risk assessment and referrals. A key finding is that 73% of women under 49 who develop cancer in 10 years have no family history, the main criterion in NICE's approach. The study suggests multifactorial risk assessment could be more effective, but would increase resource use, referrals, and false positives, prompting discussions on how to implement it safely, equitably, and cost-effectively in routine care. Boadicea, especially with polygenic scores, improves calibration and discrimination modestly. But removing polygenic scores sharply reduces sensitivity and increases the number needed to assess for broad population screening. Authors note that broader risk assessment would heighten resource demand and false positives, emphasizing a balance among practicality, cost, and potential harms and benefits. NICE issued a cautious statement, saying it is open to future updates as more feasibility and outcome data become available. Experts highlight the trade-offs between ease of implementation, accuracy, and costs in revising risk criteria. Public sentiment shows openness to proactive risk assessment for 30 to 34-year-olds and to invitations for risk assessment and use of tools like Boadicea, despite the prospect of more assessments and genetic testing. Beyond risk tools, the study underscores general cancer risk reduction measures and prompt GP consultation for unusual changes to support earlier diagnosis. A newly identified carnivorous plant challenges what we thought about flowers. Saxifraga candelabrum, found at high elevations in the Qinghai Tibet Plateau and Hengduan Mountains, actively attracts, captures, and digests insects, absorbing their nutrients and providing direct evidence of carnivory in this genus. This discovery expands the known environments where carnivory occurs in flowering plants and supports Darwin's century-old ideas. Field tests show the plant emits scents to lure prey, indicating an active prey attraction strategy rather than incidental capture. Researchers conducted feeding experiments with nitrogen-rich prey and traced nitrogen uptake to confirm the plant absorbs nutrients from captured insects, not just traps them. Independent experts say the finding is significant and note its potential for agricultural uses through transferring carnivory-related genes to crops for natural pest control. The prey-to-predator transition is described as a striking adaptive innovation, though the exact evolutionary pathways remain unclear and warrant further study. Experts emphasize that carnivory coexists with pollination, as larger pollinators are not trapped, allowing feeding and reproduction to proceed. The discovery highlights ongoing botanical exploration and diversity of carnivory, prompting reassessment of plant nutrition and ecology. Lead researcher Hung Sun notes that Saxifraga candelabram's simple traps reveal a wider spectrum of carnivory strategies among plants, challenging stereotypes about carnivorous species. The work continues Darwin's observational approach, validating historical suspicions with modern methods and underscoring the value of early natural history insights. Scientists Sun, Soltis, and Myers describe the finding as novel and significant, noting that carnivory may have evolved convergently across multiple plant families as new examples emerge. A breakthrough in gene editing is opening new possibilities for allergy sufferers who want a dog. A scientist and colleagues used CRISPR to create two beagles, Bailey and Alfie, whose saliva and fur lacked the main allergy-causing protein. Skin prick tests showed no reaction to the edited dog, while the non-edited beagle triggered an allergic response in the test subject. Published in the CRISPR journal, the researchers described this as the first successful gene-edited hypoallergenic dogs, with potential to extend to other breeds and service animals. Experts caution that safety and efficacy must be evaluated across more people, and ethical considerations must be addressed before wide adoption. Future work aims to test the approach on additional dogs and service animals to determine broader applicability and safety across diverse populations. Regulatory and safety checks were conducted, including off-target analyses, and all animal research took place in licensed facilities under veterinary supervision. While this advances the possibility of broader pet access for allergy sufferers, it is not a universal solution and may not apply to all dog-related allergies or breeds. Long-term health effects need study, and commercialization could raise costs, regulatory hurdles, and potential impacts on shelter dog adoption. Commercializing hypoallergenic dogs is expected to take years, with ongoing health monitoring and questions about effects on pet adoption dynamics. Ethical and industry caveats emphasize practical health, welfare, and regulatory oversight over cosmetic applications, and ongoing veterinary oversight will be required for the dog's health and well-being, even as genetic tweaks appear to be rare and not immediately harmful. Aging reveals a protein called EPS8 that becomes more active and promotes toxic protein aggregation linked to neurodegenerative diseases like Huntington's and ALS. In aging worm models, researchers showed that knocking down EPS-8 or its RAC partners stopped these toxic aggregates and protected neurons, establishing a causal link in that system. Human cell models of Huntington's and ALS mirrored these results. Lowering EPS-8 levels provided protective effects, suggesting the mechanism is conserved and potentially relevant to people. Scientists caution families not to alter treatments or pursue unproven interventions based on these findings, and advise consulting neurologists for experimental considerations while using clinicaltrials.gov and patient organizations to locate trials. For clinical relevance, the results must be confirmed in mammalian nervous systems, with careful safety and tolerability assessments for targeting EPS-8 signaling before any drug development, which could take a decade or more. The study underscores the value of simpler model organisms in uncovering disease mechanisms that may translate to humans, though this work is mechanistic laboratory research with no human participants. It does not prove EPS-8 drives disease in humans, nor that reducing EPS-8 is safe or druggable, and the exact mechanism by which EPS-8 activation leads to aggregation remains to be clarified. Clinically, the findings point to a shared aging-related pathway as a potential therapeutic target, but there is no immediate clinical application and standard care remains unchanged. Overall, the work advances understanding of how aging contributes to neurodegenerative diseases and points toward new avenues for treatment development. A regulatory mechanism was identified. The ubiquitinating enzyme USP4 controls EPS8 stability. Reducing USP4 lowers EPS-8 accumulation, extends worm lifespan, and mitigates disease-related changes, with USP4 knockdown in human ALS mutant cells preventing neurodegeneration. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.